Okay, thank you, Francis, for this introduction. So I'm Elisa Arnaud. I recently joined FODE as a swine technical manager. But before this experience, I was a PhD student in Chagask and Southeast Technological University, supervised by Dr. Padar Laulo, Gillian Gardiner, Torres Sweeney, and Professor John O'Doherty. And today I'm going to talk about some of my thesis results, which was entitled Pre- and Post-Weaning Nutritional and Management Strategies to Increase Piglet Growth and Age. So just to give you a little bit of context, in commercial farm, pigs are weaned around 3 to 4 weeks of age. And this is a very challenging time for piglets because they are changing of accommodation. More often, litters are mixed and they change of diets, moving from the sow milk to a new dry pelleted diet based on some plants, cereals, etc. And this often leads to a drop of feed intake. I'm just going to do this. This often leads to a drop of feed intake, and we can say that this happened at the worst time for the pig, because as you can see on this graph, first of all, the maternal immunity is decreasing around this time, and the pig hasn't built its own immunity. So this happens during this critical window where the immunity is weak. And in addition, at this time, the level of lactase, which is an enzyme necessary to digest the sow milk, is still high, and all the level of the other enzymes, which are necessary to digest this new feed, are low. So because of this, post-weaning, we often experienced a growth check, so a reduction of growth, and we often see the apparition of post-weaning diarrhea. So until now, it was common in farm to use a pharmacological dose of zinc oxide during the 2 first weeks post-weaning to prevent this growth check and the apparition of post-weaning diarrhea. And it was also possible to use in-feed antibiotics for prophylactic group treatment. However, due to the emergence of antimicrobial resistance, it is now prohibited to use pharmacological dose of zinc oxide and also to apply group treatment in pigs. So this is the first problem. On another hand, you know that the sow have been selected for their prolificity. And as you can see on the graph on this slide, the number of piglets born alive significantly increased over the last decade, from around 12 piglets in 2010 to around 15 piglets in 2023. So this means that there is more and more piglets to raise for the sow, and not all of these piglets have access to teeth. And this is critical because we know that a pig needs to receive at least 200 grams of colostrum within the first 24 to 48 hours of his life to survive. So this is now difficult to achieve in this large litter, and because the sow is still producing the same quantity of milk and colostrum, meaning that there is less colostrum and milk available per pig. And we know that a decrease in milk intake can negatively affect weight gain and weaning weight. This is also very bad, as a higher weaning weight is correlated to better lifetime performance. So we need solution to raise large litter, increase weaning weight, and facilitate weaning transition, which also can limit antibiotic usage in piglets. So the overall objective of my thesis was to find strategies to increase intake and growth of the pig in the first few days post-weaning, with a view to increase lifetime growth in pigs and reduce the reliance for antimicrobial usage. So to answer to this objective, we first conducted a small literature review, and we observed that we could act at different stages of the pig life. We could act very early in the life of the piglet when the pig is just born by trying to increase the colostrum intake and therefore trying to build more robust pig and act on its growth very early on the pig life. And we selected two strategies, split suckling. So this is when you removed a part of the litter to give access to the other part, to the udder and then to the colostrum and milk. And these strategies in the literature, we saw that it was promising in terms of growth and mortality of the piglets when it was applied during the first day of life. We also saw that applying an analgesia to the sow postpartum could show promising result on growth and immunity transfer from the sow to the piglets. But we did not know from the literature if this strategy were efficient in increasing colostrum intake and then reducing the reliance on antimicrobial usage. So that was the first stage on which we could act. On another hand, you can also act during the lactation by trying to better prepare the pig for the weaning transition and increasing its feed intake early post-weaning. And in commercial farm, it's commonly accepted to creep feed the piglets. So you provide a solid feed when the pig is still under the sow, and this has been shown to prevent the intake drop at weaning and it's also a good strategy to adapt its intestinal structure to the dietary change that it will face at weaning. However, the result on growth and intestinal health varies a lot depending on the creep intake, so the actual percentage of piglet eating this creep feed. And we know that liquid creep feed can be a solution to increase dry matter intake pre-weaning, but we don't know if this strategy can also increase intake and growth post-weaning and consequently act on antimicrobial usage. And the last thing could be to try to act very early post-weaning by increasing post-weaning feed and nutrient intake. And from the literature, we saw very good results in some trial where people try to supplement pigs with milk or liquid feed during the first week post-weaning. So there was positive effect on a dry matter intake and on growth. But we don't know yet its effect on antimicrobial usage. And we wonder, could this liquid feeding post-weaning be a vehicle to provide novel feed additive? And we selected one in particular, the glutamine, which is a fuel for the enterocyte in the epithelial barrier. It has been shown to improve the intestinal structure, but we don't know yet also if supplementing glutamine post-weaning can help in terms of antibiotic reduction. And more importantly, most of these strategies had been tested on the pig for a couple of weeks, but we did not have any data about their effect on the lifetime growth of the pig. So this literature review has been published as an article, as a review in Animals, so it's available if you want to have a look. So from this review, we selected a couple of strategies that can be applied pre-weaning or post-weaning, and this is what I'm going to present today. So first of all, pre-weaning intervention. So the first trial we did in Chagask had for objective to investigate if the application of split suckling with or without postpartum provision of an NSAID drug to the sow could positively impact the colostrum intake in suckling pigs and their lifetime growth. So this trial was a 2x2 factorial design, with the factor being application of split suckling, yes or no, and provision of meloxicam postpartum, yes or no. So regarding split suckling, we decided to remove the 6 heaviest piglets for 1 hour. These piglets were placed in a bottomless box on the heat pad next to the farrowing crate, and then they were placed back with the sow for 1 hour, and this procedure was repeated once. Regarding the application of meloxicam, we injected a product called loxicam, which contained this active molecule, meloxicam, and this was done within the 2 first hours post-farrowing. We recorded the individual pig weight, the colostrum intake, and the antibiotic and anti-inflammatory usage, and all the data were analyzed by SAS. So what did we observe? In terms of growth performance pre-weaning, we had an interaction effect between split suckling application and meloxicam provision on the average daily gain of the pig and on body weight. And basically, what happened was that when split suckling was applied, there was no improvement in average daily gain. But when split suckling was not applied, there was an improvement in average daily gain for the treatment analgesia. And we had the same interaction effect for body weight. So split suckling was cancelling the effect of the provision of analgesia. If we have a look at the main effect on body weight, so on the first line, you have presented the effect of meloxicam on piglet body weight at day 6, 14, and 27 days of age. And on the second line, there is the effect of split suckling on piglet's body weight, similarly at day 6, 14, and day 27 of life. So for meloxicam, we observed an increase in body weight when piglets were raised under the sow which received the meloxicam post-farrowing compared to control sow. And this was maintained at day 14 and 27, and we observed an increase in weaning weight of around 350 grams. For split suckling, on the contrary, so there was no significant effect at day 6, but at day 14 and 27, we observed a decrease in body weight, leading to a reduction of weaning of average weight by 200 grams at weaning. And when we had a look at the data in detail, we observed that actually applying split suckling was detrimental for the piglets which were removed, and this led to this decrease in body weight. So the decrease of body weight was mainly due to the removal of the piglets. We also had a look at the colostrum intake, and to do so, we used the equation from Tail et al. 2014, which takes into account the weight gain, the body weight of the piglet at birth, and the duration of colostrum suckling. So we used 24 hours. And we observed that piglets raised under sow which received meloxicam tended to have a higher colostrum intake than control piglets. We also observed a decrease in clinical cases of disease pre-weaning for those piglets and a tendency to decrease antibiotic and anti-inflammatory usage pre-weaning. So as I said at the beginning, our objective was also to follow the pig through their whole life to see if our strategy could impact the final body weight, the body weight at slaughter and the carcass weight. And interestingly, we observed that piglets raised under a sow which had received meloxicam also had a higher body weight at slaughter, and this was transferred into a higher carcass weight by around plus 3 kg at slaughter. For split suckling, we observed a reduction in body weight at slaughter, and there was no difference in carcass weight. So finally, what can we conclude? Meloxicam increased the body weight of piglet at weaning. Split suckling decreased the body weight of piglet at weaning. Maybe this approach was too aggressive, the way we applied it in this trial. We observed a reduction in clinical cases of disease and the tendency to increase colostrum intake in pig from the sow which received meloxicam postpartum. The benefits in terms of body weight at weaning was transferred at slaughter for piglets raised under sow which received meloxicam. And today we can say that applying meloxicam is a very inexpensive strategy and it's not very labor intensive. So it's very interesting to use this strategy on farms. So finally, we could recommend to provide meloxicam to sow postpartum as soon as possible on the delivery of placenta. Just to give you a short example, we used to do economic calculation in order to see if our treatments were economically sustainable. And for €1 invested in meloxicam in this trial, you could aim to use €55. So this is massive. It's just to show that this is a very inexpensive strategy, and there is nothing to lose to provide meloxicam to the sow in your farm. So this was a very early strategy that you can apply in the pig life. Now, I said at the beginning of this webinar that we can also act during the lactation by providing a dry or a liquid creep feed. So we did a second trial which had for objective to determine the effect of providing supplementary dry pelleted starter, a liquid milk replacer, or a liquid mixture of milk replacer and starter diet as a creep feed to suckling pigs in order to observe if this strategy could help to increase the growth, to maintain the health, and reduce medicinal usage in pigs. So this trial had 4 treatments. A first controlled treatment in which no creep feed was provided. A second treatment in which a dry pelleted starter was provided as a creep feed. A third treatment in which a liquid milk replacer was provided as creep feed, and a fourth treatment in which a mixture of liquid milk replacer and liquid starter was provided as a creep feed. The dry pelleted starter, DPS, was provided to the pig from day 10 of age until weaning, and the liquid milk replacer and liquid mixture were provided from day 3 of age to weaning in order to mimic what was really happening in Irish farms. So the liquid milk replacer, it was a commercial formula mixed at 100 grams of dry matter per liter of water. And for the liquid mixture, you can see on this graph what we did. So we started by feeding piglets 100% liquid milk replacer from day 3 of age to day 6. And then we decreased the percentage of liquid milk replacer and we increased the percentage of liquid milk mixture in the creep feed until reaching 100% of liquid feed during the last week before weaning. Both liquid creep feed were provided via the automatic milk delivery system, the baby feed system by shower. So as you can see on the picture on the right, we had a little trough set at the head of the sow, and this is where the liquid creep was provided. During this trial, we measured the individual pig weight. We followed the creep feed disappearance, the antibiotic and anti-inflammatory usage, and we also slaughtered pigs post-weaning in order to assess the intestinal morphology of the pig. So in this trial, in terms of pre-weaning result, we measured the body weight of the pig. There was no effect before day 19 of age. At day 19 of age, it was still a bit confusing. We could see a tendency for the liquid milk replacer to increase body weight compared to the dry pelleted starter and the liquid mixture, but nothing was really clear. But at weaning, we could see clear differences with an increase in weaning weight for piglets which received the liquid milk replacer compared to the control and the dry pelleted starter, and also an increase in weaning weight for piglets which received the dry pelleted starter in creep feed compared to the control. For the liquid mixture, there was no significant difference. Which was interesting is that we followed the dry matter intake of the pig during this trial, the pre-weaning creep feed intake, and we could see a higher dry matter intake for the pig supplemented with the dry starter compared to the liquid milk replacer. But we think that this was due to wastage during this trial because it was not followed by better performance compared to the liquid milk replacer, for example. As I said during the material and method, we slaughtered pigs at day 5 post-weaning in order to study the intestinal morphology. So there was no treatment difference on villus 8, crib depth, and the villus 8 to crib depth ratio in the duodenum and jejunum of the pig. However, we observed significant difference in the ileum, with a tendency for the dry pelleted starter and the liquid mixture to increase villus 8 in the ileum compared to the liquid milk replacer and the control. We also had a numerical increase in terms of villus 8 to crib depth ratio for the free crib feed treatment compared to the control. We followed the growth also post-weaning. Unfortunately, for the liquid milk replacer, we observed a reduction in average daily gain and gain to feed from weaning to day 6 post-weaning, and there was no difference after day 6 post-weaning for any of the treatment. There was no effect on carcass weight as well, and no effect on clinical cases of disease or medicinal treatment usage. So to conclude, by feeding a dry pelleted starter and a liquid milk replacer, we were able to increase weaning weight by 200 grams and 400 grams. There was a numerical increase for the mixture, but nothing significant. Unfortunately, we did not observe any benefits of our crib feeding strategy at slaughter. Liquid milk replacer decreased average daily gain and gain to feed early post-weaning, possibly explaining the lack of post-weaning growth response to slaughter. Dry pelleted starter and the liquid mixture tended to increase villus 8 in the ileum, and there was no effect of therapeutic treatment usage. So I also wanted to highlight that today milk powder is very expensive, and we did just small economic calculation on feeding cost. So it cost us around €1.41 to feed this liquid milk replacer per pig during the lactation, which is almost 3 times the price of the normal dry pelleted starter that you would feed in your unit. And all the interest also of trying to feed a liquid mixture of milk powder and liquid feed is to reduce the cost. As you see, we were able to reduce the cost by 2 by feeding this liquid mixture. So there is no lifetime benefit of crib feeding, but pre-weaning growth was substantially increased by supplementing a liquid milk replacer. So just for you to know, we also did a second trial, which was designed by one of my colleagues, Shivaza, and where we changed the setup of the automatic milk feeding system. And in this trial, Shiv observed a benefit actually of feeding the liquid mixture. So if there is an interest, this has been, it's going to be published, and it has been published as an abstract at the last EAAP. So this is all for the pre-weaning strategy. Now we can also act early post-weaning. And we conducted a last trial, which had for objective to determine the effect of post-weaning supplementary liquid milk replacer and or dietary inclusion of 1% L-glutamine on the lifetime growth, health, and medicinal usage in pigs. So this trial was again a 2x2 factorial design trial, factor being provision of liquid milk post-weaning, yes or no, and provision of L-glutamine, yes or no. So the supplementary liquid milk was provided from weaning until day 10 post-weaning, and the L-glutamine was added at 1% in the starter diet and or in the milk powder. So as you can see on the picture of the right, piglets, they had access to supplementary liquid milk on the top of their normal diet. So they were not only fed the supplementary liquid milk. In this trial, we measured the individual pig intake per pen. We followed the individual pig weight. We analyzed the intestinal morphology, and we took jejunum stripping for gene expression analysis at day 7 post-weaning. We also collected feces at day 11, 49, and 124 post-weaning in order to follow the evolution of the microbiome. So in terms of result, there was no interaction between glutamine and liquid milk replacer. Glutamine had no effect on growth, diarrhea, or medication usage, and no effect on intestinal structure at day 7 post-weaning. However, supplementing a liquid milk replacer increased the dry matter intake from weaning to day 10 post-weaning. And this is a massive increase because we were able to double the dry matter intake. Piglets which were only fed the dry pelleted starter had around 240 grams per day of DMI, while piglets supplemented with the milk powder had around 447 grams per day of DMI. This led to an increase of growth up to slaughter and an increase of carcass weight by plus 2.6 kg. We also observed an increase in villus 8 in the duodenum, jejunum, and ileum of the pigs at day 7 post-weaning, but there was no effect on medication usage. So we followed also the microbiome of the pig during this trial. There was no effect on alpha diversity. However, we saw some effect on beta diversity. So on this first graph, you can see that our fecal sample were clustering by day, first of all. And when we had a look more in detail, we saw that at day 11 post-weaning, so at the end of the supplementation period, the feces collected on pigs which received milk replacer clustered separately from the feces collected on pigs which did not receive milk replacer. And when we had a look at the difference in terms of genera, we saw that this was mainly due to an increase in Oscillospiraceae, UCG2, and Rickenelaceae, RC9, for the pig which received the milk replacer, which is interesting because these two genera, they are short-chain fatty acid producer, so we can imagine that this was beneficial to the pig. We also had a look at the gene expression at day 7 post-weaning in the jejunum of the pig, and we could see a decrease in the expression of genes coding for pro-inflammatory cytokines, so IL-17, 18, and 22. And we could also see a decrease in the expression of gene coding from small peptide transporter. And this is particularly interesting because these peptide transporter are also correlated to the setup of inflammation. So we could imagine that feeding the milk replacer also decreased the intestinal inflammation globally in the pig. So to conclude, there was no effect of supplemented piglets with 1% glutamine post-weaning. We observed an increase in carcass weight by plus 2.6 kg in pigs supplemented with milk for 10 days post-weaning. Supplementary milk increased villus 8 in the duodenum, jejunum, and ileum of the pig. It decreased the expression of gene coding for interleukin, pro-inflammatory cytokine. And supplementary milk also increased the abundance of beneficial bacteria in the feces of pigs at day 11 post-weaning. However, as I said, for the previous trial, the milk powder is very expensive, and we calculated that it would have costed €4 more to supplement pigs with this milk replacer for 10 days in comparison to the control treatment. And this is today not affordable in farms. So we could recommend to provide the milk replacer up to pigs up to 10 days post-weaning, but future trials should investigate how to optimize the supplementation period and to feed maybe a mixture of feed and milk instead of just a milk replacer in order to fine-tune the economical cost. So in terms of costs, just also for your own interest, we calculated €4 extra cost for the treatment dry pelleted starter plus liquid milk replacer in comparison to the dry pelleted starter alone. And we did an economical simulation to see if we could have a benefit by reducing the period of supplementation from 10 days to 4 days. And Unfortunately, we still have an extra cost of 3.5 euro because we don't observe an increase in carcass weight by supplementing pigs for only 4 days. And this was a trial done also by one of my colleagues, Shivvaza, which is now published in Animal. So overall conclusion, unfortunately, in our condition, it was detrimental to apply split suckling to piglets. Meloxicam provision to sow is an effective and economically viable means of improving colostrum intake and growth in piglets and decreasing clinical cases of disease. There is a benefit of supplementing liquid milk powder pre-weaning on intake, growth, and weaning, but there is no lifetime benefit, and the milk powder is very costly. There is a potential of feeding a liquid feed to pigs early post-weaning on their growth and intestinal health, but it's still too costly. There is some limitation of the use of L-glutamine in liquid feeds, maybe due to bacterial fermentation. And today, the use of milk powder is expensive and must be economically justified. For your time and listening to this webinar, and I would like to acknowledge also the partner and founder of this work in Chagask, and also all the student, technician, farm staff from the Chagask pig department and SCTU laboratories for their help in conducting the different experiments. Thank you. I'll take any question if there is.
Go ahead, Clemence.
Hi, Clemence. I can't hear you.
Clemence, I think your mic is off. No. Okay.
I think we have a...
Yeah, another question. Yeah. I think he's still typing it out, but do you mind if I ask you my question as well?
Oh, please go ahead, Francis.
One of the things that I noted in your study is that you said that split suckling doesn't really work in your experimental conditions.
Yes.
I just wanted to make sure to understand how you did it, because I've seen some papers and talked to some colleagues who said that this is like the future of suckling piglets, that you really need to, that they're really positive about this. I was just wondering what you think happened in your experiment that you weren't successful in using this.
Yes, I think it was too intensive in our experiment. So in terms of methodology, we decided to remove the piglet twice for 1 hour and a half. And this means that there was a lot of interaction with the pig very early at the onset of the farrowing process, because we started also split suckling 4 hours after the onset of parturition, so 4 hours after the birth of the first piglets. And to remove twice these piglets, this means that we enter into the farrowing crate 4 times. And we also managed them a lot. We handled them a lot because we had to mark them on the back. So I think that this was detrimental. Also because maybe there were too many handling of piglets due to experimental condition also.
Okay.
So I'd say maybe just 1 time removing the piglets would be enough, not 2 times, and definitely not more than 1 hour and a half.
Okay, just a follow-up for that. Is this so in typical commercial farms, what do? They just remove them once for 30 minutes like this?
So that's also the point. it can vary a lot from farm to farm. So we try to give advice for the Irish farm, but you would have people removing piglets maybe the day after parturition. So their aim is always to get more colostrum into those piglets. So they won't, in general, do it, and they won't advise to do it above 24 hours after birth. But it can vary a lot from farm to farm, depending on belief and also depending on the availability on the staff on farm.
Okay, thanks. And I see some chat in the questions in the chat. Do you mind if I read this? So once,
Oh, yes.
so if they, I think Bertrand, you're available to.
Yes. So I can see some questions. So yeah, can you resume the L-glutamine role? So the L-glutamine role, we decided to supplement L-glutamine because this is a very important amino acid for the maintenance of the intestinal barrier. It is used by intestinal epithelial cells as a fuel, so as a source of energy for these cells to function properly. You also have to know that glutamine, free L-glutamine, is normally present in the sow milk, and its concentration in the sow milk, the concentration of free L-glutamine, increases during the lactation, which means that often we can consider that at weaning, there is a shortage of this amino acid for piglets, even if it's not an essential amino acid. So that's why we decided to supplement L-glutamine, and this is the role that it can have in the gut. I hope it answers to your question. Yeah. I'm looking at the other question in the chat. How can you explain the beneficial effect of meloxicam exactly? So meloxicam, it's a non-steroidal anti-inflammatory drug. It's an anti-inflammatory drug which there are some studies which demonstrated that it can be transferred from the sow to the piglets when it's given intramuscularly. So we believe that maybe meloxicam was transferred first of all from the sow to the piglet, so it could have also some anti-inflammatory effects early in the life of the pig, explaining the resultant clinical cases of disease and on the tendency to reduce medication usage in piglets. We also believe that, farrowing, it's quite a painful time for the sow. So if you help the sow to overcome this period, if you help the sow to recover better, then the sow will be better able to nurse her piglets. And if she is better able to nurse her piglets, the piglets are going to get additional colostrum intake. We were able to demonstrate an increase in colostrum intake in this trial when the sow had received meloxicam. And then this means that piglets can also have like a better transfer of immunity and then a better like growth. I hope this answers to your question, Bertrand. Last question, can we extrapolate your thesis to data at 21 days of weaning piglets performance? I'd say this is a tricky one because 1 week of weaning age can make huge difference. So what I can say for sure is that it's working at 28 days of age. But if it's even more challenging, in my mind, then there is even more need to these alternative strategies, and this should I think the result should be you could extrapolate the results at a 21 of age. I know that in the US, yes, this is the common weaning day. In Europe, we tend to the age of the pig at weaning, but yeah. not standard in all the countries. And Clémence, you said, did you see any effect on the different diets on the behavior? And we did not really record the behavior of piglets. The only thing we recorded was the feeding behavior, so the percentage of eaters and non-eaters. And what was interesting in this study, so for example, we recorded the percentage of eaters and non-eaters during the second trial. So the trial in which we supplemented piglets with either a dry crib feed or a liquid crib feed. And we observed that piglets supplemented with dry crib feed, they had a higher number of eaters compared to liquid crib feed. So once again, I think that they were mainly playing with this crib feed and there was a lot of wastage because this was not translated in a better growth up to weaning. So if your piglet eats, the growth must be, I mean, the feed must be transferred somewhere. So if there is no transfer in terms of growth, it means that there is certainly some wastage, unfortunately. You're welcome, Bertrand. Talk to you later. Okay. Okay. Is there any other question?
I think that's it. Okay, yeah. So again, like everyone has been telling you in the chat, thank you, Elisa. It's the presentation was really, really good and it was really interesting. Congrats on this work.
Thank you for your time, and don't hesitate to reach out to me if you have any other question regarding this presentation. Thank you.